Hello, everyone. Welcome to the first EABU session of the spring. Thank you for joining us. I'm Sheila. I work at MSU for the Department of Entomology. So today we're going to take a closer look at a relatively new but increasingly important invasive species affecting our forests here in Michigan, the elongate hemlock scale. This pest was first discovered in our state in 2010, which was quite a leap from its previously known range in the eastern United States, and it's raised some serious concerns about potential impacts here in the upper Midwest. We're joined today by Toby Petrice, a research entomologist with the USDA Forest Service Northern Research Station. Toby focuses on the ecology and management of invasive forest insects, and today he'll be walking us through recent work conducted along the Lake Michigan coast, looking at elongate hemlock scale's impacts on eastern hemlock and surveying for its natural enemies. As always, we'll have time for questions at the end of the presentation, so feel free to drop your questions in the chat at any point. And at the end, I will read them out and we'll get to as many as we can during that Q&A session. So with that, I'll turn it over to Toby.
Okay, so thanks for the introduction, Sheila, and thanks everybody for joining today. Uh, so today I will be talking about elongate hemlock scale, a newish invasive pest of hemlock trees in Michigan. And like Sheila said, I'm a research entomologist with the Northern Research Station of USDA Forest Service. We're actually stationed here in East Lansing, Michigan, and I'm next door to Sheila, and I just met her, so she's in the building next door to me. Okay, so elongate hemlock scale's actually been in the U.S. for a long time. It was first discovered in the eastern U.S. around 1908. It spread throughout most of the northeastern states and then down into the southern Appalachians. In the northeastern states, it's primarily on hemlock, but as you go southward, you find it more and more on actually Fraser fir Christmas trees, and that's how this thing moves around a lot. So it's moving around on cut Christmas trees, and then also on ornaments that are made from live Christmas tree boughs. Now, it was first discovered along the coast of Lake Michigan around 2010 during hemlock woolly adelgid surveys. And because this was a pretty significant jump from its previously known distribution in the U.S., there were some concerns on what impact this species might have in the upper Midwest. So damage from the insect, so it is a sap-feeding insect that feeds on the mesophyll tissue of the needles, and it can cause needle yellowing and stippling. Needle cast, if needle cast is severe, it could cause branch dieback. Reduces tree vigor. It's been reported in the literature that heavy infestations may also kill trees. It is an oligophagous species, so it attacks several different conifer species. Up to 62 have been reported in the literature in 13 different genera. Vanette et al. Reviewed the literature on hemlock, or well, I'm sorry, elongate hemlock scale natural or host range, and he found that it primarily is on hemlock, and it does some aesthetic damage to Fraser firs, and those are the main host of the species. The other conifer species it attacks, it generally does not survive well on them, and if it does survive, it usually doesn't produce many offspring. So elongate hemlock scale is the typical armored scale. So armored scales secrete this hardened covering over their bodies to protect them. And it starts out in the crawler stage. So these are less than a millimeter long hatchlings that crawl around. They have legs and antennae, and they either crawl around or they're blown around by the wind or splashed by rain droplets. But this is the main mobile stage, at least for the females. Once they find a suitable host, they'll insert their mouth parts into the leaf tissue or needle tissue. They'll start absorbing their antennae and legs, and they'll secrete this waxy covering over their bodies. Once the first instar is mature, it'll molt into the second instar, and this is where it elongates from underneath the larval skin of the first instar. And as it elongates, it will be secreting a new covering over its body. From the second instar, we can either have a mature female, so she will stay under that covering of the second instar, and that covering will harden and darken. Or if it's a male, it's actually very different in appearance. So smaller in size, and they have a different color of waxy covering. It's really bright white, and this is what you often notice when you see a tree infested with the elongate hemlock scale, because these males really stick out, and the covering is really brittle as well. Now, the male, interestingly, will become a winged adult, and then it will fly around and find females. It will mate with the females, and then the females will produce these little eggs underneath that hardened covering. So, because of the new find of elongate hemlock scale in Michigan, and how highly valued hemlock trees are along the coast, there's some really high value real estate there. People really cherish their hemlock trees, but also it plays an important role in bank stabilization. So it's growing on these sandy dunes. We started some work back in 2020, actually, during the COVID, looking at some aspects of the insect's phenology and its impacts. And we picked two locations in Southwest Michigan where populations of elongate hemlock scale were really high. And if those of you know, we also have hemlock woolly adelgid in Michigan. So we selected sites that did not have hemlock woolly adelgid so that we wouldn't have to worry about active treatments occurring for hemlock woolly adelgid, but then also to see what impact the scale itself was having on the trees, rather than the scale and hemlock woolly adelgid. So in 2021, we got a full year of data on the phenology of the scale, and what we found was that so across the x-axis here we have the collection date, and then the proportion of individuals on the y-axis, and then this line shows the proportion of scales with eggs, so females with eggs. And you can see that the females with eggs are most abundant in the winter months, and they decrease until summer, and then they increase again going into the fall. So the point here is that even though they tend to be highest in the winter, they're still present throughout the growing season. And the same with the crawlers. So we found that they peak in late spring or early summer, but still present pretty much throughout the season. This results in a couple things. So first, we found that most of them are overwintering as adult females or adult females with eggs. And then, of course, the crawlers peak in early summer. But because these crawlers are present throughout the season, it results in these overlapping generations. Part of this is because the crawlers are actually quite abundant before the new shoots elongate. So that relegates the crawlers to colonizing actually older needles. So if you're looking at an infested tree, you'll almost always find higher numbers of scale on the older needles, because they keep building up on the older shoots, because they don't have new shoots to colonize when they first start hatching. So this figure here shows the densities of scale. So on the y-axis we have the mean scales per 100 needles, and then we have year across the x-axis, and then white bars are for scale densities on current year needles, and green bars are the previous year needles, so 1 year prior. And you can see that trend where the previous year needle densities are always higher, whether relative or significant, than the densities on the current year needles. But then also going from year 2020 to 2023, we see this uh, increasing trend in scale densities. Uh, in part, this is probably related to having these mild winters we've had, letting the scales build up. They can be sensitive to cold winter temperatures, especially the younger instars, which we find that some of them do successfully overwinter. Most of the time, it's the females that successfully survive the winter, but also some of the younger instars appear to be surviving the winter, and resulting in some of the population actually having 2 generations per year. We also wanted to look at the impacts of the scale on hemlock growth. So we used methods that were developed actually for hemlock woolly adelgid, and we sampled branch tips from trees. We collected 4 branch tips per tree, and then on each branch tip, we measured the length of the new shoot growth on the 5 longest shoots. And then we compared that to the densities of scales on the individual trees. And you can see for 2 of the 4 years, so 2021 and 2023, we did find a negative correlation with scale densities and shoot growth. 2020 and 2022, we didn't see a significant difference. I don't know why it wasn't consistent. I'm guessing that part of it is because of other factors such as rainfall affecting hemlock growth, and there's some kind of interaction going on there. To look at impacts, we also looked at canopy transparency. So this was basically transparency is a measure of basically of the amount of foliage or needles missing from the tree. And we estimated this on the trees in 5 different plots in 2020. And then we went in 2024, we reevaluated all these trees. And what we found is actually, even though some of the trees looked really unhealthy, not much of a change or no change in canopy transparency, except for this one plot in plot 10 in the center. If we look at it, like we overlay the scale densities here, so this brown line is the scale relative scale densities. You can see that plot 10 did indeed have the highest scale densities, which may explain the increase in transparency. But if we look just to the right at plot 11, not seeing much of a change in scale density or in transparency, even though scale densities were very high. So this tells us that it they're definitely causing loss of needles to the trees, but it's a slow process. It's going to take several years to actually be able, even more than the 4 years of the study, to actually measure the changes in tree health. So if it is killing these trees eventually, it's probably taking over a decade to actually kill the trees. I should also note that at none of these sites or in the area we found any mortality related to the scale, or that we could attribute it to the scale. We also wanted to look at the natural enemies present at these sites, and we found this species in Carcia citrina quite abundant. So this is a parasitoid that attacks several different scale species. It's described as a cosmopolitan species, so it occurs throughout most of the northern hemisphere, and I'm not sure. I think it even occurs in the southern hemisphere. It attacks not only the armored scales, but it also attacks a lot of hemipterans. It's reported as being a very important natural enemy of elongate hemlock scale in Japan. And it's pretty easy to determine that a scale has been parasitized by Encarsia citrina, or any scale parasitoid for that matter, because the adults will chew this little round exit hole to exit the scale covering. So we looked at seasonal parasitism by this species. So this figure, we have the collection date along the x-axis, and then the proportion parasitized along this left-hand y-axis. And if we look at parasitism rates on the previous year needles, we can see that from going from winter to early summer, really not much of a change. And this is because we're measuring parasitism from the previous year. So it's just residual parasitism, because these scales will persist for several years on the needles. So we're just looking at parasitism from the last year. Then it decreases because we have abundance of younger instars building up here. And then we see it shoot up going from mid to late July and increasing in through the fall months. And we see a similar trend here where the new shoots about early July, we start seeing scales become mature enough to be attacked by the parasitoid. And after that, it just increases dramatically throughout the fall. Now, this species of parasitoid prefers the second instar scales. So if you remember this little picture of the second instars, and if we look at the abundance of the second instar scales, we can see that parasitism starts to increase dramatically right after we see this peak in second instars present on the needles. I'm guessing they prefer these second instars because the covering is probably thinner and easier for them to penetrate with their ovipositor. Overall parasitism for the year 2021 was about 40%. So pretty good parasitism. If we look at parasitism over the 4 years, again, we have current year needles in white and previous year in green. And we have the proportion of females parasitized on the y-axis. You can see that 2020 to 2021, not much of a change. And then we saw a decrease in parasitism on current year needles in 2022. And then this was sort of a carryover and resulted in lower parasitism rates on the previous year needles in 2023. And then also note the trend here where parasitism tends to be higher on the previous year needles. This is in part due to us measuring some of the parasitism rates from the previous year rather than the current year, because again, the needles will persist, or the scales will persist on the needles, and then the scales with exit holes we're counting that are from the previous year. So pretty good parasitism. But if we remember from the previous couple of slides before, we're still seeing the population increase. So even though we have high parasitism rates, the parasitoids are not keeping the scale from increasing in numbers. I'm sure they're having an impact on it, but they're not stopping it from increasing in numbers. We also wanted to look for predators, so we collected beet sheet samples from at least from several trees in the plots, and we did this weekly in 2021. Also, we did some in 2020, and we found, what we would expect to find: the twice-stabbed lady beetle, Asian lady beetle, brown lacewing, sort of generalist predators. But the most interesting find was this little species here called the scale picnic beetle. Its scientific name is Cybocephalus niponicus. It's in its own family, Cybocephalidae. And this thing was quite abundant. It's a tiny little beetle. It's only a little over 1 millimeter long, but when they fall into the beet sheets, we thought they were seeds at first because they're round and they roll. And then we realized they're alive. We're like, whoa, what is this thing? So we actually had a friend of mine identify it, and turns out that it's this scale picnic beetle, which was actually introduced into the U.S. a couple decades ago. And it's native to Southeast Asia, but it was introduced originally in the East and then a couple other states, Florida and Texas, for controlling euonymus scale. And it is a generalist predator on several different scale species. But in the East, they found it feeding on elongate hemlock scale as well. They moved it around to a couple places, I think in Pennsylvania and New Jersey. They didn't really do a lot of follow-ups on it. I think they confirmed it was established, but they didn't really do a lot of work looking at the impacts of this predator on the scale. But the interesting story here is that there was a an aphis lab in the southwest corner of Michigan, and they were rearing natural enemies of various species, but this was one of them. They released it into the local area for euonymus scale. Uh, I don't think they followed up to see if it did establish, and part of that was because the lab was ultimately closed and they were moved to other locations. But I was giving this talk uh, maybe a couple years ago and talking about finding this beetle, and somebody in the audience chimed in and said, "Hey, I worked in that lab. We're the ones that released it." So he was very excited to hear that it migrated up to up the coast of Michigan, and it found our elongate hemlock scale and was feeding on it there. And also, it's a interesting. It has the males have this yellow pronotum, and the females are all black. But the males look like little minions, and we would watch them feed in dishes, and they're pretty cool. Though, if they catch a crawler, they'll actually roll over on their back and sort of hold it in their legs and chew on it. Looks like a sea otter feeding or something. So again, this beetle species is very abundant at the sites, and it was very concentrated on trees with high scale densities. So this shows this figure shows the scale density across the x-axis, and then the number of Cybocephalus niponicus recovered on the y-axis. And you can see that they're really targeting those trees with high scale densities. Uh, Looking at the seasonal abundance of the species in 2021, the brown line shows the mean number of adults recovered, and the gray line is the larvae. You can, if we look at the brown line for the adults, you can see that the adults are present almost any time when it's warm enough. But they tend to peak, or they did peak in the late summer and fall of 2021, where the numbers got quite high. So they must be overwintering as adults in the leaf litter, and when it gets warm enough, they're just crawling up the trees and starting to feed on the scales. And we also found the larvae from early summer to out through late fall. Uh, I'm not sure how many generations the beetle is having in these locations. I'm guessing it probably has two generations at least, given the length of time we found the larvae in the on the collected in the beat sheet samples. We wanted to see how many scales this beetle could feed on, and then the stages it preferred. So we did some little laboratory assays where we put three female scales, or three female beetles in a petri dish, and then three males in the dish, and we repeated this several times. We fed them a sort of a smorgasbord of scales, if you wish. We gave them first instars, second instars, and adult females all at the same time, and they really tended to like the first instars. Part of this is probably because it's a smaller food source, so they're eating more of them, and they tended to feed less on the adult females. It probably takes more effort for them to chew their way into the over or through the scale covering to reach the female. So they probably this suggests that they do prefer the younger instars to feed on. And like I mentioned earlier, they also feed on the crawlers. Okay, so for different controls of chemical controls of elongate hemlock scale. Now, Our group really, we didn't try to any chemical controls, but just looking through the literature, there's several products that have been used. Dinoteffuron has listed with inconsistent results, let's say. So some results say it works fairly well, and this is most of this work is in Christmas tree plantations or in nurseries or in arboretums. They tend to report better control actually with the synthetic pyrethroids, also insect growth regulators. They Some growers are combining a couple different chemicals to control it, and then also some are using horticultural oil. Uh, just remember that when you're using some of these contact insecticides, you're going to have to apply them multiple times if you want to get really good control of the insect, because this those overlapping generations where you have crawlers emerging throughout the growing season. Also, some things to consider. Excuse me. If you're treating forest trees as canopy height, so you're going to, in this situation, some kind of systemic is going to be your best choice because you're just not going to be able to reach the canopy foliage. You also have those overlapping generations to deal with. Uh, you don't want to harm the natural enemies. So are systemics better? Well, if we find one that works, systemics would be the best option. Then also, I'm guessing that the parasitoid would be less impacted by uh, contact insecticides because that scale covering is going to protect the parasitoid developing under the scale covering. But the beetle, Cybocephalus, is going to be present throughout the whole growing season, so it's probably going to come in contact with insecticide. And then also you have to consider wetlands and riparian areas. So I feel like this does need some more effort to determine a good systemic that would work for elongate hemlock scale. There is some anecdotal evidence where some of the hemlock woolly adelgid treatments are also reducing elongate hemlock scale that are also on the trees, but it's anecdotal and it probably deserves like a planned research study to figure out a good systemic that will work. Uh, so the potential distribution of elongate hemlock scale. So Rob Vanette and Brian Akama have been working on this for several years, and they have a new postdoc, Clarissa Moore with the University of Minnesota. Brian Akama is with the University of Minnesota. And Rob Vanette is with my research station, the Northern Research Station. But they found that the super cooling point of elongate hemlock scale was actually negative 27. But if it had prolonged exposure to negative 20C for 7 days, 50% would die. If it had exposure for at negative 20C for 23 days, 95% would die. And these maps show the likelihood of reaching these cold temperatures in different regions. And if we so the first column is showing consecutive days when it's negative 20 for either 7 days or 23 days, and this is the right hand column is showing non-consecutive days. Then also this bluish green area is showing the distribution of hemlock in North America. And just to simplify the map for you, the darker the shading, the higher number of years we're going to see these lethal temperatures. And looking at these maps, it looks like the scale will be able to survive at least some winters throughout the distribution of elongate hemlock scale in North America. But what probably is going to help us with this insect is when we get those polar vortexes where we have these sudden super cold snaps, and that is probably going to help the most with knocking back the population. And then I'm almost finished here. I just wanted to show you this slide of a different scale species that we found on hemlock. when we started working on this, it was a bit confusing. We didn't know if we were looking at elongate hemlock scale or another scale. But on the left, of course, we have elongate hemlock scale. And remember that always has this skin of the first instar at its apex. So that's a good way to identify it if you're looking at the females, especially. Another common species we found was black pine leaf scale on hemlocks. It has the same coloration as the elongate hemlock scale. It's usually more oval rather than elongated, but some of them do look elongated. So it can get confusing for some people. But if you look close and don't see that pupil scale or pupil skin, or I'm sorry, first instar larval skin at the apex, then it's probably not elongate hemlock scale. Another species we found was pine needle scale on hemlocks. Now, the males of elongate hemlock scale resemble pine needle scale, but if you if you do find something that you're unsure of, then you would want to inspect and see if you find the females present of elongate hemlock scale. If you're not finding them, you're probably looking at pine needle scale. And then the last one is cryptomeria scale. So this one resembles elongate hemlock scale younger instars, like the second instar when it's elongating. Sometimes it looks similar to those, but again, you would not find this first instar larval skin at the apex of it. And this is a cool little scale. It really does look like a fried egg. So it has this sort of whitish clear covering with this bright yellow body underneath that looks like an egg yolk. This is another invasive species. It was new to Michigan as well. Not nearly as abundant as elongate hemlock scale at those sites, but it still is there, and we'll probably continue to monitor that one as well. Okay, just to wrap things up, so we did find that elongate hemlock scale could reduce shoot growth for some years if densities are really high. The parasitoids and predators were abundant, but they still weren't stopping the scale population from increasing. We probably need a cold snap to knock them down that way.
Hang on. Okay, I got rid of him. Okay.
Uh, and then we didn't really find a significant change in transparency, even though some of these trees look very unhealthy. But it takes a long time apparently for the trees to die from the scale, if they actually ever die. And then we didn't observe any mortality at these sites. It's definitely not as a lethal pest,
Uh,
hemlock woolly adelgid, and it's likely to establish throughout the distribution of eastern hemlock, according to the cold tolerance studies. And if
so
it
you have S's and Sam,
someone's not muted, someone someone's not muted. And then finally, chemical control may be necessary for heavily infested trees, especially if they're high ecological or aesthetic value. Like I mentioned before, there's some really high value properties along the coast of Lake Michigan, and they really cherish their hemlocks. And some people want to know, what they can do to make their hemlock trees look more healthy. And then just want to thank everybody that helped with this work, all the Forest Service technicians and MSU technicians and students. Lots of people over the 4 years of the study. They did all the hard work. Some of it was fun, some of it was not so fun, but just a big thanks to all of them. And then also the funding from, of course, my station, the Northern Research Station, but then also state and private and tribal forestry contributed most of the money for this project, along with Michigan State University, and then field sites from Michigan DNR, and then two private landowners, Tim Horwitz, and a private club, the Prairie Club. And with that, I'm finished. I can take any questions you may have.
We can wait just a minute and see
Oh,
any questions coming in.
thank you for saying something because my screen here is trying to get the screen back up.
Someone asked, is this something that is spreading rapidly?
No, it seems to be spreading very slow. We had some nearby sites where we surveyed for it, and about it it they were probably a couple kilometers away, and we finally found it there this year. So it seems to spread really slow. Even though the males are winged, the females are not. So the only way that they can actually move is the females can move is at the crawler stage, and then also because they do reproduce sexually, it does slow down movement because you have to have both sexes present. Let's say if something's wind blown and you only have one crawler that's blown, 100 meters away or something, a male is going to have to find that female crawler after she matures. So.
Any other questions? I'm not seeing. Oh, there we go. Will this be coming south into Indiana?
Uh, if it's right, it's really close to the border now, and it might already be there. Wherever there are hemlocks, it has the potential to spread there and establish there.
And then someone said, would you have a suggestion for ideal timing of systemic application, such as dinotiferon? Oh, I butchered that, but based
Yeah.
on phenology.
Uh, so with a systemic, I think that actually timing is less important for the insect itself, but more important for the phenology of the tree. So usually systemics are applied in the spring and early summer. That's when most of the sap is moving and when they're translocating the most. Uh, but for hemlock trees, I'm not sure. You might have a wider window. I think they have a wider window for treating for HWA, but I'm not positive of that. But I know for treating like, for example, hardwood trees, it's more it's important to treat them in the spring and early summer.
Any other questions? Okay. Well, then I think that we can wrap it up. Thank you everyone for joining us today. I want to say that our next EABU session about emerald ash borer will be Tuesday, May 13th at 11 a.m. So you can register for that on our website, and I will drop a link in the chat so that everyone has that. Um, so you can get more information about future Emerald Ash Borer University sessions at that link and register for any that you're interested in attending. And thank you, Toby, for your presentation.
Thank you, Sheila, and thanks everybody for coming. Hope you enjoyed it. If you have any questions, you can reach out to me. You could just simply Google my name and you probably get my email address. I'd be happy to answer them.